So we are now going to start on chapter 17. So there's a big, jump from 4 to 17 because we want to go with the flow. Now we want to talk about cell reproduction and differentiation. So chapter 17 fits in that. So don't be surprised, we're not going to chapter 5, it's chapter 17. Okay. So the outline of chapter 17, from 17.1 to 17.3 we're going to study, okay? So that's the topics we're going to cover in this chapter. First we're going to look at the cell cycle, so how it creates new cells. Cells get damaged, you need to replace them. So the cell cycle is very important for that. Organisms grow, they need new cells, so the cell cycle is important for that. Regeneration of tissue, repair of tissue. So cell cycle is very important for many of those functions. So 7.1 covers that, and then I'm not going to go through the specifics of it, but we will cover that in the lecture as we go through. And 7.2 we'll look at the structure of the DNA and its function. So it's a brief look at it. We're not going to look deeply into it, all right? So briefly. So there's many topics covered in that. I'm not going to go through that. 7.3 we will look at the reproduction, how one cell becomes two. Remember, all cells come from pre-existing cells.
Yeah.
So cell reproduction is important for that. Many topics covered in there. Let's just get into it. So as usual, the objectives of the lecture. This slide is very important for your benefit. Okay. So after we finish this chapter, you should come here and ask yourself these questions or these objectives. See if you know this information, if you learned it. If you haven't, then you need to go back and revise. Okay, so I'll just give you an example. So you should be able to summarize the activities of each phase of the cell cycle. Okay, you don't know it now, but you will, inshallah. What the stages are and what their significance is. Okay, so basically this is the significance of the objectives. All right, let's get on to lesson 1, the chromosomes. Okay. So cells, we say, reproduce by dividing into two. So you have an existing cell that divides and creates a identical cell to the original one, two identical cells. Okay. So for the purpose of unicellular organisms, this is actually the mechanism of reproduction. So unicellular cells, remember, one cell, like bacteria, like some protists. Okay. So when they divide, they're actually reproducing themselves. So cell reproduction is important, cell division is important for the reproduction in these organisms. While in multicellular organisms like us, cell division is important for tissue repair, for growth, for regeneration, okay, and those sort of mechanisms, okay? So, when you start off as a fertilized egg, as the first cell, when the sperm and the egg join and form the zygote, so this is the original cell, the first cell is called a zygote. After that point, cell reproduction creates the different tissues, the different cells in your body. Okay, so we just studied histology, we got different tissues in our body. So cell reproduction was important because from that one cell, that stem cell, okay, all these different tissues arise, okay? So we use it for growth and change, okay? Now in eukaryotes, the mechanism cell division is the same in all the eukaryotes, okay? So if we have a look at the structure of the DNA, because remember the DNA controls the activities of the cell. So it's very important that the new cell has an exact copy of that DNA. So we just need to cover what the DNA looks like. So this is what double-stranded helix. So DNA actually is what we call a double-stranded helix. Now let me see if there's a image here. Yeah, right here. So this is what this double-stranded helix is referring to. This sort of structure, and you've seen one in the foyer down the bottom.
Yeah.
See that DNA?
Yes,
So it looks like this. Is like that. That's what the helix structure is referring to. It's actually 2 strands of DNA. Now, I'm not sure. I think we studied this in chemistry of life. Remember the nucleotides?
Yeah.
ICTG. So that's the code for DNA, okay? Now, when we look at DNA, so one strand will have one code, and then we have on the other strand what we call the complementary code. So there's base pairing between nucleotides. So I always base pairs with T and C with G, okay? So the double strand I'm talking about, so on one strand will be this, on the other strand, the complementary. So that's what makes the double-stranded helix, okay? So this intertwine, this is what it's talking about. Intertwine into one another, all right? That's the classic double-stranded helix of the DNA. So we find that during normal cell activity, that the DNA is found in this structure called chromatin, okay? This grainy substance, you know. I'm not sure if there's an image. So this is what chromatin looks like that, okay? That's an actual electron. What happened to our wiper? Don't have one? Do you mind if you go to that other classroom and get one, please? Thank you. So chromatin exists in this form, this grainy form, okay? During normal cell activity, okay? Because the cell needs access to the DNA to perform its normal functions. So it has to be this relaxed form where it can access the DNA code to make proteins and so on, messenger RNAs, and you know. So this form, this grainy form here, is known as chromatin, okay? And usually when you look at, thank you. When you look under the microscope, so you find that you can't see the DNA. It's not visible, all right? Under normal light microscopy, which we will do, you know, in the lab. You will see some onion cells in different phases of the cell cycle, and actually you can see the chromosomes, okay? So DNA in the form of chromosomes is visible under the light microscope, but it's not visible in the form of chromatin, okay? So in this form it's not visible. So it's only visible when it forms chromosomes, okay? So these sort of structures, okay? So what happens is, during cell division, the DNA needs to be compacted, so it's easily separated during the division into the two new cells, okay? So it forms these nice units called chromosomes, and in humans we have 23 pairs of chromosomes, okay, that we need to separate. So only during cell division does these structures appear, these chromosomes, okay? So what it is basically making the chromatin into a compacted form, which forms these chromosomes, okay? Okay, so the chromatin, as I said, is a relaxed form of the DNA, and we find that it's made up of DNA and protein, okay? So this protein called histone is very important for providing that structure where the DNA wounds around, okay? Now, if we see here, this is actually the histone proteins right there, and the DNA is wound around. So if you have a look at the histone, you know, it's like that, and then DNA is wound around it like that. All right? Well, I'm not an artist, a very good artist, but that's what it looks like, okay? So that's what chromatin is. So DNA around histone proteins, and there's actually 8 of them, okay? So this doesn't show it properly here, but if you have a look at this figure, you can see that there is 8 histone proteins, okay? So it's a 3D image, there's 8, okay? There, 8 histones, and the DNA wraps around that. That is known as this thing called a nucleosome, okay? So when DNA wraps around the 8 histones, it forms the nucleosome, all right? Any questions so far? Yeah.
What's the bigger, the chromatin or chromosomes?
So chromosomes obviously is much larger. Well, what do you mean is bigger? It's made from.
That's the more general.
Well, chromatin doesn't exist as a grainy form. Chromosome is a compressed form of the chromatin. So, in terms of what you're talking about, larger. It's the same material compressed.
Which one carry the DNA?
Both.
Both of them.
Yeah. It's just, you know, you got a piece of string here, and then you grip that string, and then you go coil it up. Okay. That's what it is. You compress it, so you can easily separate it during cell division. So you got the chromatin, and then you got the chromosomes. Okay. That's the difference.
The chromatin is the single one, and when it's
So chromatin is referring to the whole DNA here, okay? That single unit is known as the nucleosome, okay? Where it wraps around the histone, okay?
No. The one pair of chromosome, it's called chromatin, and both of them it's called chromosome.
Well, in the form when it's relaxed, it's called chromatin. When it's compacted, it's called chromosome, okay?
Okay.
Basic. Okay. That's actually an electron micrograph of DNA wrapped around the histone. So that each one of those is a nucleosome there. We have to, you know, this. I'm not sure if you read the part on the microscopes in chapter 3, I think it was.
It's not included.
It wasn't included, but if you had read it, we have light microscopy, which it has a maximum magnification of 1,000. And these electrons, they can go to much higher, like 100,000. That's why you can see DNA. You can't see this with a light microscope. All right, so chromatin forms the chromosomes. So chromosomes are actually, what we said is compacted forms of chromatin. And it only appears during cell division, okay? So when the cell is about to divide, it forms these structures, these chromosomes, all right? Now, our chromosomes are, each chromosome consists of one unduplicated chromosome, okay? So during cell division, okay, the DNA is replicated because you want to end up with a copy of the DNA in each cell. To do that, you need to replicate the DNA. So DNA replication happens. So an exact copy of the chromosome is made, okay? And this forms what we call a duplicated chromosome. And the duplicated chromosomes have 2 sister chromatids that are identical. So these, you know, here you got one unduplicated chromosome. So when you duplicate it, it forms 2 sister chromatids, we call them, okay, that are joined together at the point where we call the centromere.
It's separate or it's copy itself?
It's joined to. What do you mean it's copy itself?
When it's replicates,
Yes.
it's copy itself or it's separate?
First separate, after that.
See,
No, it's copy.
this chromosome is in the form of chromatin, okay?
Yeah.
During that phase, when it's in the chromatin, what we call the S phase of the cycle, DNA is replicated by proteins within the nucleus, okay? What we call polymerases. It makes a new copy of the DNA during that time. And then it forms these duplicated chromosomes during cell division, okay? So they're joined here at the centromere, these what we call sister chromatids. These 2 are identical, okay? And this is what's separated during the cell division into the new. So during division, each cell will get one unduplicated chromosome. Okay? So then the cycle returns back, you get cell division, a cycle. So you understand what these terms mean, unduplicated? Duplicate means a copy, okay? In this case, it doesn't have a copy, so it's called unduplicated. When you get replication, it becomes duplicated, okay? And the duplicate chromosomes are called two sister chromatids, okay? So we got the sister chromatids joined at this point, which is called the centromere. And this centromere holds these together until you get the point where they're separated. Okay, this is very important. Okay? So, as I mentioned, during the S phase, we haven't talked about that yet, but the cell cycle has what we call a synthesis phase, an S phase, where DNA is replicated. So each chromosome becomes two molecules, which we call it duplicated chromosome. Obviously, it's a copy of itself, so it's a duplicate. And as I said, it forms the two sister chromatids, which are held at the centromere. So basically, before the cell division, you got unduplicated, you get DNA replication, then we form these two sister chromatids joined at the centromere, then it becomes one duplicated chromosome, and then you get unduplicated, right? So in humans, we have 46 chromosomes. So this happens to each one of those before cell division, okay? So, as I mentioned, the sister chromatids are identical to each other, because the reason is you want to end up with the same DNA in the new cell. So every cell in your body has the same DNA as the original cell that you came from, the zygote. So it's identical, okay? The only cells that don't have the same DNA is the sperm or the gametes, we say, okay? Sperm and egg. They'll have different DNA to the original cell. So they're identical and they contain the same genes. So the same genes are carried on each sister chromatid, and that's important for DNA, for cell division to form two new identical cells. Okay. All right. So we talk about the chromosome number. All right. Now the chromosome number is not the same in all the species. Yes.
So you have a sperm, we call the egg ova.
Ova or ovum, okay? Together they call the gametes, okay? Okay, the sex cells, alright? Gametes, okay?
Both?
Gametes together. So, the chromosome number is not the same in all species, Humans, monkeys, trees, and so on, we have different chromosome numbers, okay? So we have, but individuals of the same species, they always have the same. So I have the same number, you do, some, other humans, all humans have the same, okay, number of chromosomes in male and female, all right? And they come in what we call homologous pairs, okay? Homologous pairs means that you derived one of the same chromosome, for example, chromosome number 1 from the father, okay? So what we call the paternal chromosomes, okay? So paternal means from the father. And one came from the maternal chromosome, the mother. Because during sexual reproduction, when the sperm and the egg come together, and we're going to talk about this later, about haploid and diploid cells, okay? This will make it much easier to understand what I'm talking about. The sex cells have half the number of chromosomes, okay? Compared to normal cells.
Yeah.
So when they come together, it restores the full number of chromosomes. That's why they come in homologous pairs, okay? Because you get one from the father and one from the mother, okay? So that's what homologous pairs mean. So here, this actually shows the human chromosome number. We have 23 pairs. You see, each chromosome comes in pair. Can you see that?
Yes.
Even the sex chromosome. Okay? So in females, the sex chromosome is also homologous. But in male, it's not. Okay? It's two different chromosomes, X and Y. Have you studied that
Yes,
before?
yes.
Okay?
And bonus XX.
XX is homologous. So in female it's 23 homologous pairs. In male is 22 homologous pairs in one sex, X and Y. Okay? So you see this pair, one of this pair. So during fertilization, I'll just draw this pair. Okay? Chromosome number 1. So chromosome number 1. Okay? You got one of it in the egg and one of it in the sperm. Okay?
23.
So 23, yes. So there's 23 single chromosomes. They're not in pairs. When these join together, it's fertilized. You get the zygote. You get 46 or 23 pairs.
Yeah.
Like this. So that chromosome number 1 and that join together to form a pair. Okay?
Doctor.
So that pair, you see, it came from the mother and the father. That's why they called homologous chromosomes.
Doctor, in the sperm there are 23 pairs of chromosome.
No, 23 single.
23 single
Yeah.
chromosome.
Yes.
In one sperm.
Yes.
So they become 46.
We will learn about this in the next slide, okay?
It's not one chromosome and one chromosome after that it duplicated.
No.
23.
23. You'll learn about gametogenesis later on in the chapter, okay? But for now, just understand that these homologous chromosomes, where the origin is from, I'm trying to explain that.
Okay, yeah,
The
yeah.
origin is from that, okay?
From mother and father.
From the mother and father. So, if you have a look at these, we can see, you see, we're looking at them under the microscope, they look nearly identical to each other, okay? So they have the same copies of the same genes, each one of those, but in slight different versions, okay? And this is important later on when we study genetics, okay? When we're talking about dominant and recessive, this is very important for that, okay? Because some, your mother has a feature, characteristic, and your father doesn't.
Yeah.
And then you don't have it, okay? Because it might be recessive. The gene that your mother gave you might,
not be there.
have it, and your father doesn't have it, but because it's recessive, it's not showing in
Not
you,
showing.
okay?
The phenotype and genotype.
This phenotype is masked. And this is because there's different versions of the same gene on each chromosome, okay?
Yeah.
That's what it means here, okay? So it has the same genes, but in different versions, okay? And that is important when we study genetics, okay? You understand that?
And doctor, genes they mean by alleles or?
No, genes.
Well, so alleles, you find this allele, and then there's the gene, okay? The genes actually form the, the allele. All right. So as I mentioned, we have diploid and haploid number of chromosomes. So the difference between is in normal cells, what we call somatic cells, okay? See, somatic cells are every other cell in your body except for the sperm and the egg, okay?
Sexual cells.
Sexual cells, okay? The gametes. So this is the term gametes I talked to you about before. That's what it means. And this gonads is referring to the testicles and the ovaries, all right? So testicles in the female, ovaries in, testicles in the male, okay? We don't find many females with testicles, okay? That was a good one, huh? All right. You better cut that for the YouTube. All right. So Let's try that again. We got the gonads is referring to the ovaries in the female
Yeah.
and the testicles in the male. This is where the haploid cells are made, the sperm and the egg. Okay, so somatic cells are all other cells except for the gametes. Okay,
Yeah.
and in this diploid we find that
Two N.
we find a pair of homologous chromosomes like this. You see, as I mentioned, see chromosome number 1. There's one from the mother and one from the father. Okay, so it comes in homologous pairs. So to understand this better, we got this naming for it. If it's diploid, we give it this
Two N.
2N. Okay, so 1N from the mother,
One with the father.
one from the father. So 2N in this case, what's the number of chromosomes is in human? So 23. 2 times 23. Okay,
Yeah. 46.
46. Okay, so that's what it means.
Yeah.
So each chromosome is as a homologous pair in diploid cells.
Yeah.
Okay,
Mm.
so in this case, N from the mother, N from the father. So in humans, 23, 23. Okay,
Yeah.
which gives the 2N. 2 times N, 46. Okay.
Yeah.
Now in haploid cells, as I mentioned, we find all this
Sperm and egg.
in the gametes, in the sperm and egg, which are made in the gonads, as I said, is the ovaries and the testes. All right. The N number, okay, because it has half the number of chromosomes. Okay.
Yeah.
Because during gametogenesis, what we call the formation of the sperm and the egg, what happens is that the cells lose when they divide, there's what we call what happens is that duplicated chromosomes divide.
Okay.
In the first meiosis, okay, so there's 2 meiosis, and in the second meiosis, wait, do I get that right? So what happens is that during meiosis, you get unduplicated chromosome pairs separating. So these homologous chromosomes, what I'm saying, they separate, okay? So what happens is you got the cell, okay, the original cell. So chromosome number 1, it has the homologous pair, okay?
Okay.
Now, during the formation of sperm or egg, you get, let's talk about sperm because it's easier. Egg, there's some funny things happening where, you only form one cell and 3 cells, what we call polar bodies. They don't form functional gametes. But in sperm, you get 4 functional gametes. So what happens is, when you get cell division of the gametes to form the sperm, this this homologous chromosome pairs, they separate during the cell division. Okay? So you end up with 1 chromosome in each of the sperm. Okay?
1 versus 2.
Alright? So you started with 2N and now you end up with 1N. So N. So that's why we have haploid. Now the reason we got haploid is, now later on, when humans reproduce by sexual reproduction, so this is from the father,
come from egg.
this is from the mother, the same thing happened there, okay? One of the chromosomes, okay? So this is N as well. When these two join together to form the zygote,
It becomes 2N.
it becomes 2N. If we don't do that, it will be a disaster for reproduction, okay?
4N.
You'll have 4N, it'll be
So many diseases.
chaos, okay? Diseases, everything. So it's very important that we need to halve the number of chromosomes. So we start with 2N, we halve it, and then during fertilization we return back.
Okay.
So does that make sense?
Yeah.
Yes.
Any questions?
Here.
All right. So let's have a look at the human cell. Actually, this photo, I should say, that you see here is what is called a karyotype. Okay, it's used to detect certain diseases. You know how you see these different colors?
Yeah.
You can actually detect changes in the chromosome structure, because you can see differences in the colors. You can detect certain diseases. Okay, so in the molecular biology lab, they will use this to detect a disease of the chromosomes. Okay, like Down syndrome. Okay, Down syndrome, you have an extra pair of chromosomes, number 21, I think. Yes.
Yeah.
So you will see 3 chromosomes for 21. So they can detect that. And there's other genetic problems. What happens is you get deletions of chromosomes, duplication of regions, and so on. Okay. So this karyotype is very important for detecting diseases. Okay.
Doctor,
Yes.
if there are maybe 2X and 1Y, the person will be man and woman at the same time.
We're gonna study that later on. Okay.
But what it be 2X and 1Y or?
Turner or line?
I think there's a Turner syndrome, or
Turner,
there's
yeah.
another syndrome there. You're gonna take that, okay? In the end of the, you get, I forgot which disease it was,
There's
okay?
disease, yeah.
There is one where you have one X, there's one where you have one,
XXY the
one Y, I think, and where you have 2 X's and a Y, like this. There's combinations of it. Okay, and this is because we get unequal division of the chromosomes.
Yeah. The
Okay,
problem with the division.
unequal division. It happens. All right, in humans, as I said, we got 46 chromosomes that come in 23 pairs. So
Yeah.
you got 23 pairs here,
Yeah.
and when you count them, it's 46. Okay.
Okay.
So this is talking about the somatic cell. So it's 2N. Okay. It's diploid or haploid.
Diploid.
Diploid, okay? So the normal chromosomes, other than the sex, we call them autosomes. So the 22 pairs are known as the autosomes. So except for the sex chromosomes, the rest are known as autosomes, okay? So 44 chromosomes out of the 46. So we find that in the male and female. But when we look at the sex chromosomes, it's different in male and female, okay? So in the sex chromosomes, we have one pair, okay? XX, okay? So here it's homologous. Remember that, okay? In the male, it's XY, it's 2 different chromosomes, okay? It's non-homologous. So,
How?
in the past, before genetics, people thought that the female determined the sex of the child, and they used to, kill them or whatever, But we know now that with modern genetics, that the male actually determines the gender, because, the female only can give XX, remember?
Yes.
In this egg. The male, he can give an X or a Y or an X, okay? So, if a sperm that is Y fertilizes an egg, the child will be
A male.
male.
Okay. Because in the female, because it's XX, because that's homologous.
Yeah, the female can only give an X chromosome, so she cannot. She has no.
Because the male
say-
is determined if it's.
The male determines the sex
Okay.
of the child, not the female. Okay, so the female is always giving an X chromosome,
Okay.
while the male there's a 50-50 chance
Yeah, X or Y.
X or Y,
Yeah.
male or female. All right.
Okay.
So-
The male from where he bring the eggs?
And Ummah.
It's in the genetic.
Okay.
It's in the genetic.
It's in the genes.
It's in the gene,
When,
yeah.
come on, you're asking this
Very
question.
well.
Don't you believe that we were created by Allah?
Yeah,
He
I
made
know,
this
but
for us.
I want to.
Where did it come from? I
He
don't know.
means
don't
X,
say it
the X
scientific
from
way.
where?
Scientific way, you do you believe in evolution?
Yeah.
Huh?
Yeah, evolution.
Yeah. Why do you believe in it?
Because
What's your
I
basis for